Hey everyone, in this chapter we're going to be going through an overview of DNA structure, and then once we know the structure of DNA, we're going to go into different ways of modeling DNA, both its structure, how it bends, and how it packs. So the first thing we want to do is to go through a recap of the energy landscapes of catalytic reactions, just some important information to carry forward from the previous topic, especially the Michaelis-Menten mechanism. We'll be using some of those terms in this lecture as well. After that, we are going to go through multiple different subsections for this chapter. We're going to first start with an introduction to DNA, just so that everyone's familiar with the terminology for the structure and function of DNA. We're going to start with DNA stability and denaturation. And of course, we're going to move on to different ways of modeling DNA. So some of the main ways of modeling it would be by a random walk or a worm-like chain model. After that, we can also look at the electrostatics of DNA. So we're going to focus on that for a bit as well. At the very end, we're going to look at combining our knowledge to see how DNA folds and packs within itself. So let's go through a quick recap first. Just going to go through the key points from the previous topic. So first we learned what an enzyme was. It's basically a protein that helps to speed up energetically favorable reactions, and they help to produce disorder in the universe. So enzymes are all going to have an active site that binds to substrate molecules, and then that action is going to reduce the activation energy of a chemical reaction, and therefore it's going to catalyze that reaction and make it speed up. So over here we have two diagrams where this is originally an uncatalyzed reaction pathway where there is a required activation energy in order for the reaction to proceed. By adding an enzyme to the whole process, it allows this activation energy to be reduced, and therefore making it easier for the reactant to go through with the reaction to create the product. We then took a look at the free energy G of a system. So in the past we were kind of iffy about whether free energy was Helmholtz free energy or Gibbs free energy. In this case we're looking at G, Gibbs free energy, for most of these reactions. So in biochemistry, the free energy is a quantity that expresses the disorder of the universe, and the change in G is going to be critical in determining whether a reaction is energetically favorable or unfavorable. So the most important part for this is that, to remember from this, is that energetically favorable reactions will have negative delta G. So you want a negative change in free energy in order for a reaction to be exothermic or energetically favorable. So then the next thing that was important to understand is what happens if you do have a positive delta G. If you have a positive change in free energy, then it's energetically unfavorable, but it doesn't mean that it's not going to happen. You can always couple reactions together where there's going to be a very favorable reaction with a large negative delta G coupled with a positive delta G reaction, which allows it to move forward with that unfavorable reaction. So the key thing to keep in mind if you were to be tested on this is that the overall change in delta G is going to be, or the change in G is going to be negative. Next we took a look at some free energy landscapes. So combining them all together, we looked at a few of these separately. First, we have the simplest one, where a substrate goes through an activation barrier and then proceeds to forming some product. But if you add in an enzyme, there are certain different things that occur. So there's going to be an interaction between the enzyme and substrate, a deformation energy between the two elements, and then you have this combined energy landscape where it lowers the activation energy for the reaction to proceed. So here's a clearer diagram. You have your substrates, there's binding, catalysis, so then you form a transition state over here, and that transition state is much lower than the original uncatalyzed reaction, and then it forms your products. So apart from looking at the conceptual viewpoint of enzymes, we then took a look at reaction rates. So we had these different rate constants, K plus for the forward reaction, K minus for the backwards reaction rate, and then we had R for the rate of product being produced. And so when we had this, we were able to figure out different ways of representing your change in enzyme by adding up all of the different rate constants, forwards or backwards, and subtracting or adding them. So we were able to figure out the rate of change of each of these concentrations. So this is something that you should be able to derive yourself. We also looked at some conditions in order to lead us to the Michaelis-Menten equation. So these conditions I'll explain in a bit. So the Michaelis-Menten model basically defines the reaction rate. We have V being the reaction rate, being equal to the rate of product being produced, and that's equal to this expression over here, where you have the maximum reaction velocity times some sort of substrate concentration divided by the Michaelis constant plus the substrate concentration. So looking at the steady state here in pink, it's going to be the steady state is going to be right in the middle when things aren't changing too much. If you take a look, you'll see that over here in the middle, there's a plateau for the enzyme substrate complex. So therefore we call this a steady state. So then we have to find a couple of conditions for the Michaelis-Menten model. We have two approximations that we have to adhere by. We have the equilibrium approximation, which states that the value of R is equal to or approximately equal to 0 because it's very small to begin with anyways. And using R equals 0, we can try to find an equation that allows us to graph reaction velocity versus substrate concentration. Next, the steady state approximation states that the change in the enzyme substrate concentration is going to be equal to 0. So that represents the plateau of the graph that we just saw. So moving forward, the two most important things is really just they're really just being able to recognize and apply the Michaelis-Menten equation. So just looking at these graphs here and having the Michaelis-Menten equation defined as follows, we can determine two very important terms from a Michaelis-Menten plot. So in terms of Km, when Km increases, it means that a higher concentration of substrate is required to reach 1/2 of the maximum reaction speed, whereas the maximum reaction speed is where it's going to plateau towards. So remember, the Michaelis constant Km is going to be a measure of binding affinity of the enzyme for the substrate. The larger the Km, the smaller and the weaker the binding affinity is, because more substrate is needed in order for it to reach 1/2 of the maximum velocity. So keep in mind, you should be able to understand what the Michaelis constant represents and symbolizes conceptually, and you should also be able to figure out the Michaelis-Menten constant by looking at the graph. So apart from presenting this graph, we also presented something else called the Lineweaver-Burk plot. So that's just taking the reciprocal of the Michaelis-Menten equation to get a linear graph. And once again, you'll be able to find your Km, or Michaelis constant, as well as your Vmax. They'll just be intercepts versus looking at the graph to figure out where 1/2 of the maximum velocity is. So just briefly, we're going to go through the overview of DNA and its organization. So I want to briefly mention what the structure of DNA is, which you're probably all familiar with already, how DNA is organized, and the different ways we can model DNA. So at the end of this portion of the lecture, you should be able to recognize the different monomers of DNA, so basically nucleotides, and then be able to express different ways of being able to model DNA. And then we'll be going through multiple other sections and going into detail for each of these models. So first of all, DNA is made of nucleotides. So you're probably very familiar with this already, but DNA is made of 4 nucleotide building blocks. Each nucleotide has a sugar and a phosphate, and that's linked to a base, and that forms a nucleotide. So that nucleotide contains a sugar, a phosphate, and a base. So then there are 4 different bases that we have to be aware of. There's A, C, G, and T, which we'll go into a little bit more detail with in a bit. These different bases are covalently linked together, and the arrows here represent polarity. So there are 2 arrows here. They're going to run anti-parallel to each other in the DNA double helix, and they're going to be complementary. So we see here that the shapes and the chemical structure of the base allows hydrogen bonds to form efficiently between certain pairings. So between A and T, which we have over here, A and T, and between G and C, these shapes are going to fit perfectly with one another without perturbing the double helix, and 2 hydrogen bonds can form between adenine and thymine, and 3 hydrogen bonds can form between guanine and cytosine. So looking at the image on the right, we have just a little tiny piece of a double helix for DNA. We have a 3D structure, and it's a view from the side, and 4 base pairs are shown. So there's going to be a phosphodiester bond represented here that links nucleotides together, giving each strand a chemical polarity. So that's important. The 2 ends are going to be chemically different. The 3' end over here is going to carry an unlinked OH group. Attached to the 3' position on the sugar ring, the 5' end is going to carry a free phosphate group attached to the 5' position on the sugar ring. So just keep in mind there is a difference between the 3' and 5' end, and that those are the nucleotides being tied together by the phosphodiester bonds. And then within the double helix, you have the bases being paired together between G and C and A and T. So if we take a look at this picture, we can see a DNA double helix crudely broken down into 4 different genes. So different regions of our DNA contains different genes that code for different proteins or functions. So RNA A, coming from gene A, is going to code for protein A. Possibly gene C codes for RNA C, which doesn't produce anything. And then so on and so forth. Different proteins can be produced due to different genes in our DNA double helix. Now, in terms of DNA packing, in eukaryotic cells, very long double-stranded DNA molecules are going to be very coil-like in structure. They're so coiled, but in terms of trying to pack them up into something more delicate and more dense, you have something called chromosomes. So these chromosomes contain long strings of genes. It's a long segment of DNA that contains instructions for making proteins. So there's a very detailed folding process to get to a chromosome, which we'll be discussing throughout this topic. So just a brief overview of human chromosomes. Here's a picture in A. We have an image taken after a cell was lysed, and then in B, they were artificially lined up and paired together for easy identification. So in total, we have 23 chromosomes. So here we have, we've posed the question, how is DNA packed? So you saw that chromosome before in that picture, but how does DNA get from a double helix to that? So in terms of what's happening, your DNA double helix, it has a width of 2 nanometers, very small. And additional packing is going to occur at each level. So DNA first wraps around some histone molecules. And then those histone molecules are going to fold in within themselves to create a chromatin fiber of packed nucleosomes. So each one of these is called a nucleosome. And now you have something that's thicker. So this packed nucleosome structure is going to be approximately 30 nanometers in width. And then next, all of these are going to fold in on themselves to form these loops. And in total, that's 700 nanometers in width. And this is one subsection of your chromosome. So the important thing here is that each DNA molecule has been packaged into something called a mitotic chromosome that is 10,000-fold shorter than its fully extended length. So in order to save space, it's going to delicately fold itself into a more condensed and packed structure, and it's going to be approximately 10,000-fold shorter than its original length. So that's pretty amazing considering so much is happening on essentially the nanoscale. So this is the first electron microscopy picture that was taken of a nucleosome. So especially here, chromatin was isolated from interface nucleus, appearing as 30 nanometer thick threads. So this is kind of what this image is. When these were decondensed experimentally, the nucleosomes in B were shown. So you have DNA being wrapped around individual histones. So we call these beads on a string because it literally looks like beads on a string. So let's take a closer look at the nucleosome. You have DNA, so that's the DNA double helix wrapping itself around the histone octamer. And it's an octamer because there are 1, 2, 3, 4, 5, 6, 7, 8 pieces to the structure. So then that's just a very quick overview to DNA because if you take molecular biology or cellular biology, you're going to learn a lot more about the details, about how each of these work, but our goal is to figure out ways to model different aspects of DNA. So that's what we're going to do in the next few topics. So we can model DNA in many different ways, and we're just going to be looking at a few of these throughout this unit. In the first one, we have a sequence. That means we have information provided to us. In terms of sequence studies, you'll learn more about that in molecular bio courses. In the next portion, we have protein binding sites, which can allow us to study interactions of DNA with other molecules. Also something that's best left for a molecular bio course. The next three are going to be the focus of our next few chapters, or not chapters, but topics. In the charged rod model, we model DNA as a giant charge because it does have polarity. It's highly negatively charged. We then have a molecular spring and a random walk model, depending on the size of DNA. So we're going to be taking a look at these two as well in quite a bit of detail.